All right, so last time we were talking about kind of the basics of the membrane, and right now we're going to start discussing the major functions of the membrane proteins as they exist in this lipid bilayer. So we have transport, enzyme activity, signal transduction, cell-cell recognition, intercellular joining, and the attachment of the cytoskeleton, or attachment of these proteins to the cytoskeleton ECM to kind of hold everything together. So if we look at this, the first one, the transport proteins, this is where you have an integral or a transmembrane protein. In other words, the protein is going to span through the entire membrane, and that's what's going to allow things to come in. So you can see, if you look down here, you can see these blue dots, these red dots, etc. What they're doing is they're moving across the entire membrane through the protein. And the protein can be a shuttle, it can be a transport protein, it can just be kind of like an open port to allow things through. It doesn't really matter. And then what this does is it can be used to allow certain types of hydrophilic substances to cross the membrane, which wouldn't normally be able to do that due to the hydrophobic nature of the interior of the membrane. In other words, like right through here, for instance, this is hydrophobic. So these things could not, these hydrophilic things or these charged ions, so forth, could not be able to get through. So what that protein does is kind of makes like a little conduit or a hydrophilic channel for these things to get through. And then some of the channels allow for passive transport that's just basically moving or diffusing down a concentration gradient. And then others can use an active transport mechanism to pump things, of course, against their concentration gradient using ATP. And the sodium-potassium ATPase would be a good example of this type of a thing. So if you just kind of look at this little cartoon right here, it says, "What you got there, Jim?" And then the guy on the right here says transport proteins. So when he eats them, he's able to get through. So I just thought that was kind of a interesting little cartoon that somebody had sent me a while back because they remember learning about transport proteins in my class. So they're getting through the cell wall there. Okay? All right. So anyways, the enzyme activity, some proteins, of course, are enzymes that we know, and we will learn more about enzymes in Chapter 8. Oftentimes these things work alone. Sometimes they work together as a team to perform particular functions. They can be a transport protein to allow things to pass through. They can act in a way to kind of relay a signal from one spot on the outside of the membrane to a signal cascade on the inside. Doesn't really matter. They have catalytic types of properties, which is what an enzyme is. Okay, they can transmit signals. So, for instance, if you look here, if this is a hormone, for instance, and it binds to this protein here, and then it kind of transduces the signal or transmits the signal inside the cell, and then it sets off a series of events that can occur inside the cell, similar to with the enzymatic activity that we were talking about earlier. And then the transport protein that we talked about first, that can also have enzymatic activity if you think of it like the sodium-potassium ATPase pump. So it's acting like an enzyme to speed up the moving of the sodium and potassium ions. Okay. There are some ID tags like we saw in the video in the first part there that are made out of glycoproteins, so they're proteins with sugars attached to them, and this is an ID tag, an identification kind of molecule whereby the organism is able to tell self cells from non-self cells. So you can see here these glycoproteins. This is enabling certain cells to know that these cells belong in the organism, and other cells that have other ID tags but are not self ID tags would be recognized as foreign, and your immune system, of course, would get rid of those things. Okay, intercellular joining. Some things kind of, either link two cells together or create a channel for things to get through by linking two cells together. And we'll kind of learn as we go through and we start talking about plants and plasmodesmata and gap junctions, those types of things. What those basically are just channels for things to get from one cell to the next, and they're often done so moving through proteins. The other thing that can kind of happen is these two proteins can kind of link together right here and kind of form an anchor that really provides some structural rigidity to the actual tissue or the cells itself. So, that's a way that proteins can behave. They can attach to the extracellular matrix and some of the portions of the cytoskeleton on the inside, and then some of this extracellular matrix on the outside, and just kind of help these cells kind of maintain the shape of the tissue that it is, whatever the case may be, but oftentimes these proteins that are involved, okay? And then if we look at like a transmembrane protein, we learned about primary, secondary, tertiary, and quaternary structure early on. Well, if you look here, you can kind of see that this region right here, we have kind of an alpha helix, okay, making up kind of this portion of the protein, and then, you know, the same with all these other ones here as we go all the way around. Well, as these things are linked together, all of these alpha helices kind of form a little channel through which things can get through. So in other words, you know, you can get things moving through here because this portion, the way this thing is arranged, is kind of creating a channel that allows things to pass through, you know, into the cell, or perhaps even kind of moving from the inside to the outside of the cell, just depending on how this particular thing works. So this is just an example of how the secondary structure that we learned about in chapter 5 kind of relates to some of the stuff that we're talking about here. Okay, so that wraps up part 2. If you have any questions, make sure you write them down. We'll talk about them in class, and we'll get on with part 3 here soon. Okay, we'll talk to you soon.